So we'll go ahead and get started now. Thank you for making it out to the last Nano at Tech for the semester. Today we have the pleasure of having Yonggang Ke give us a presentation on the self-assembly of DNA-based nanomaterials and environmental remediation. Dr. Ke received his PhD from Arizona State University in 2009. From 2009 to 2014, he was a postdoctoral fellow at Harvard University. And since 2014, he's been working in the Wallace H. Coulter Department of Biomedical Engineering at Emory University and Georgia Tech. Dr.
Thank you. Thanks for the introduction. I'd like to thank you again, all you guys again for coming to this seminar. This really great environment. I think I've been here before, but never gave a talk here. Actually, it's nice to be a speaker. Actually, as you can tell from the title, my lab primarily does DNA-based nanomaterial self-assembly. You probably also noticed there's a smaller fund for environmental remediation. That's because actually organizers suggest me to put that in there. I'm not going to talk about that part, actually. Not that I don't care about environment. Actually, biomaterial, in this case, DNA actually is great material to be used as a sustainable material, which is good for environment. I'm pretty sure you can use this type of DNA-based nanomaterial for environmental protection-related research as well. But that's actually not the focus of my research, so I'm not going to talk about that part. So it's primarily, the talk is going to be about making DNA nanostructures, making DNA-based nanomaterials, and a few applications as example for you to grasp the general idea how we can use, or we use DNA for some applications. Yeah, being at my lab is actually at Emory, health research, science research building. Been there for 9 years so far. Oh, by the way, if there are any students here, I'm going to have a class next semester on this campus. I'm going to discuss DNA-based nanomaterials. So if you're interested to take the class, please look it up or send me email. I can offer you more information. So the field I work on is called DNA nanotechnology. There's a different aspect, a different flavor to this type of research. But the general idea, the most fundamental idea, is kind of just summed up in these slides. So we take DNA not as a genetic information storage molecule, like what a biological system does. We actually use DNA as a building material, right? Molecular building material. So instead of store genetic information in biology, as you all know in biology, genetic information is stored in DNA, and then that information transferred to, most time, transferred to phenotype protein to perform majority of the biological functions. But we are using DNA in a different way. We're using DNA as a molecular building block. So the sequence we design in the DNA is not used for store genetic information anymore. Instead, it store interaction rule or the information related to the final structure we want to build. That information determines or direct how DNA molecules interact with each other, and through DNA hybridization, lead to the assembly of a final nanostructure or nanodevice. So in this context, sometimes for some folks here, it might be useful just forget about genetic information in DNA, even though you can engineer the genetic information in DNA molecule for some applications. But in terms of building nanostructure or nanomaterial, to start, we can just simply think of DNA as an organic polymer, but it has sequence that you can program. And by programming sequence, you can determine how the molecule react with each other. So these are some examples of structure our lab made in last decade. As you can see, that includes some three-dimensional nanoscale objects with customer-designed 3D geometry. So for those objects, as you can tell, they have really complex, intricate geometry. The resolution of such structure can be as small as 1 or 2 nanometers. So you can design sub 100 nanometers 3D object with a 2 nanometer resolution for each voxel to build any geometry, any shape you want at that scale. Here are some other examples of, for example, polyhedral structure, polyhedral cages, and a larger two-dimensional array lattice with, again, designable feature, nanoscale features, the square lattice or honeycomb lattice. This is another example of a three-dimensional crystal structure or lattice structure. So this one is micrometer size. The building block or repeating unit is a 50 nanometer octahedral cage structure and connect together to form such three-dimensional object. I'm not going to have time to discuss with you about the design process and what software are available. I just want to tell you that there's a great effort to develop a design software, a simulation software, and a lot of effort goes into modeling, visualization, simulation of such DNA object. So here's a simple example of a small, relatively small DNA nanostructure. It contains 4 DNA double helix. You can see it's, six DNA double helix are connect together through our design. So essentially, you have all this colorful DNA strand going through between the helix, kind of connect them in a way we want, right? So this is our, of course, already finished design. This is just using a software to visualize the 3D geometry. This is before, of course, before any simulation or relaxation. This is not really the real configuration in solution, but there are also software available to simulate dynamic behavior or more accurate conformation of such structure in solution. In terms of nanomaterials, you guys are familiar with this concept. There's actually nanomaterials are everywhere. We know that in nature, of course, you have a lot of great examples. Something as small as a protein you can consider as a nanomaterial, or some larger structure such as this, nanoscale, feature on a butterfly wing, or this diatom structure exists in nature. So those nanostructure offers unique property. And of course, scientists or engineers also build our own nanomaterials using different methods. It's just a few small examples I just grabbed from internet yesterday. So here you can see a few examples, small nanoparticle, one-dimensional, two-dimensional, or three-dimensional nanomaterials with a single component or multiple component. Depending on the size, geometry, and composition, of course, this different material have different functions. So what's unique about this DNA nanostructure? Before that, I want to kind of, from my perspective, what is the general challenge when you work with nanomaterials? I think at least there's 3 major challenges from my point of view, especially when I work with our DNA nanomaterials. Of course, we want to make something as small as possible. This, in a way, you can compare that to a computer microchip, right? So by making it really small, getting to a couple nanometer resolution, our computer chip has become more and more powerful. In a way, actually, you can even consider a microchip as some kind of a nanomaterial. We typically don't consider that way, but it kind of have some similarity. You use a top-down approach to generate this really complicated structure, and that provides function. From that sense, it can be considered as a nanomaterial. So the challenge for developing a really useful nanomaterial platform has many challenges. I think there's 3 major challenges from my point of view, and those are challenges we're trying to address in our research. So the first thing, of course, we want to design, our design strategy should be able to control nanomaterials, shapes, size, resolution. We want to have the method to develop a nanomaterial to allow us to build any shape, any size, any resolution we want, of course, ideally. The second challenge to me is, can we build multi-component nanomaterials? because we want to have different functions in our final device. Here is a simple example, right? So let's say I want to build a biosensor. I want to detect a existence of DNA, RNA in solution. In order to do that, I could have a piece of DNA at the center as a biomolecule to react with my target DNA or RNA. But that's not enough. For this such a device, for example, I may want to put a carbon nanotube to connect my DNA. So that's going to be connect to my electrode for the detection eventually. So in order to realize function, a useful function, or more function at least, we want to be able to integrate, achieve integration of different nanomaterials to build such multi-component nanomaterials. That's the second challenge. The third challenge to me is how can we integrate this multi-component or single component nanomaterial to top-down fabricated devices. Again, take this biosensor as an example. If I want to build a high-density biosensor array, now I need to be able to incorporate this individual device into an microelectronic device and connect the carbon nanotube to the electrodes, right? That's not a small task. That's really hard to do. So those are the challenges we try to address in our research. So before I get to the some of the work we've done, I want to kind of briefly discuss what's unique from our perspective, what's unique about DNA. Of course, every material is unique, but what's unique for DNA? So DNA, for us, one of the most wonderful property of DNA is really fantastic for programming really complex designable nanostructure with nanometer resolution. That's due to a few intrinsic property of DNA. I'm sure you are familiar with DNA and particular double helix structure of DNA. So if we look at DNA molecule, there are a few really important property we like that lead to the design of a really complex structure. The first thing, of course, is capable of store large amount of information. Remember, we rely on self-assembly, right, or synthesis or molecular interaction to produce those kind of nanomaterials. So if you want to build really complex or intricate structure, of course, your molecule need to be able to store large amount of information. That determines, that basically limits how complex your structure can be. So DNA can store large information in its sequence. That's great. Secondly, it has really simple but robust interaction rule, molecular interaction rule. So if we look at, for example, a intramolecular interaction, a hairpin formation of DNA, or a intermolecular interaction, the hybridization between two strands, they follow this really simple Watson-Crick base pairing, right? ATGC. So it's really simple, almost like binary language, right? You have this two type of base pair, but also really robust, right? If you have different sequence, you know one side of sequence will interact with another side of sequence. You know that rule, and it's really robust. Even if you have a couple different sequence difference in your strands, that can lead to huge difference in terms of binding affinity. So that's great. The third thing is also really important is the final structure, or in this case, a typical B-form right-handed double helical structure, is very stable and highly predictable, right? So if we design a interaction such as this, we can predict what the final structure will get. So this is also important. I just give you one comparison. If you compare to this to protein structure engineering, many of you at least know it's really hard to de novo design a protein to have a structure you want. Partially is because protein have really diverse interaction and diverse structure. So it's really hard to predict. Of course, there are great progress out there with AI. New AI algorithm. The structure prediction of protein is much improved, but I don't need to lecture you. There's still a long way to go. On the other hand, this type of structure is stable and highly predictable. So what does that mean? So if you combine all this property together, what this actually means is you have a molecular language, right, sequence, can be translated directly to the final structure. So if you design a set of sequence, you can determine how they will interact. If you want to design a structure, for example, like a cube structure, you can look at this structure and look at the interaction between DNA strand and design the sequence going backward, design the sequence you want. And when you have those sequences, they will interact to form this structure. There are also additional properties that make DNA really useful. It's chemically stable, so it's really good for storing information for a long time. But on the other hand, it's also biodegradable. So it's really great for some of the applications like drug delivery or other biological-related applications. It's also relatively easy to synthesize and modify chemically or biologically. For biological synthesis, particularly, this can lead to really low energy consumption. So it's really kind of a really highly sustainable material for making nanomaterials. So those properties make it useful for information storage, other applications which we're not going to talk about today. Today's focus is going to be making really complex DNA nanostructures. So our lab primarily does three types of research. One is making DNA nanostructures, and then use DNA nanostructures to build nanomaterials, nanodevices. And being in the biomedical engineering department, we also use DNA for a range of biomedical applications. So we'll start with a short discussion about building complex DNA structures today. There are many examples, as I showed earlier. Due to time limit, today I'm just going to use one example, one type of design strategy to showcase that, to you how we can build complex structure using DNA. So this is a method developed in 2012. We use hundreds of DNA to assemble into three-dimensional structures. So we're not going to be able to cover the detailed design strategy, but it's not that complicated in principle. You just need to design these different colors of strand to interact in the fashion you want, right? So you can imagine, you can simply consider each of the strand here as a building block or a DNA Lego or DNA brick, whatever, how you want to call it. And depending on what kind of object we want to form eventually, and looking at the object, you can decide how the strands will interact with each other. And you can engineer your sequence to realize that type of interaction. What exactly the interaction will look like, we don't need to pay too much attention to that. The only thing you need to know is that we can design a strand to interact with the right neighbor, correct neighbors, right? So kind of like a Lego toy in a way, right? So if you have all this colorful piece of Lego represent the DNA strand, and you just need to design the Lego to interact with the right neighbors. And one of the nice thing about Lego, as we know, is that you can make all kinds of different shapes if you have one set of Lego. And that's also true for this DNA Lego brick or DNA brick structure. So if you have a set of DNA strands, you can actually make different shapes just by combining different strands corresponding to that particular shape. So in the end, you can design a huge number of structures in one try. So the whole process starts with an in-house design program we developed. So it allows us to kind of draw this kind of three-dimensional geometry in the software. And each dot here, you can simply consider as a DNA strands. But you don't need to pay attention to that detail. The program does that for you. So you design the geometry as you want, and that information will be translated to a set of DNA strands or DNA Lego. And then we have all the DNA strands in the 96 well place down here. Then these pipetting robots start to mix in the strand together as what the program tells it. And then we do a annealing process. So you put all the strands together in a PCR tube, and you raise temperature, all the DNA strands denature, and you lower the temperature, strands start to react with each other slowly, eventually get the structure you want. We can assemble hundreds of different designs in parallel. You just send the information to the robot. So this is kind of similar to, from design point of view at least, is equivalent to a molecular 3D printing. This is the follow-up work we did later. I'm not going to spend too much time here. The takeaway message here is that there's a lot of flexibility in such a system. We can make the structure much bigger by changing the twist, the design a little bit. So the previous structure contains up to 500 DNA strands, and this one is much larger, published 5 years later. This one contains 33,000 different DNA strands. So very expensive, as you can imagine, but that's sometimes what you need to do for research anyway. So this is a gigantic structure, contains 33,000 strands. With more strands, of course, you have higher resolution. You can build larger structure. So we showed some of the examples here. I just want to, all these, by the way, all these 3D models on top, this is just the projection view from 3 different directions. And down here, I'm sure most of you are familiar with this is a TEM image, again, corresponding to 3 different projection view. This is a teddy bear shape. This is TEM tomography. So you rotate the TEM grid, so you can look at the object from different angle, and you can reconstruct the 3-dimensional geometry. Yeah. This method is really flexible. So in order to scale up, one way to scale up, of course, to make the system larger, so each structure contains more DNA strands. Another way to make something bigger is to connect the individual structure together to make crystalline structure. So in this case, a paper published 2 years later, after the original work, we demonstrated we can make 2-dimensional crystalline structure. So you connect this building block. You can consider this blue block as one repeating DNA brick structure, and you connect them. Again, using DNA hybridization, you can make a lattice or 2-dimensional crystalline structure. And the nice thing about this system is, of course, you can decide, or you can design the thickness. You can design the nanoscale feature. For example, you can have this parallel channel. You can have periodic holes in the structure. This channel was later used, you will see an example later, to organize carbon nanotube. Carbon nanotube nanoarray. Another really interesting direction, I'm not going to spend too much time today, is to make dynamic nanostructure, dynamic nanomachines. So this is one example. Without going through details, you can see it kind of acts like a molecular domino array. What happens is that if you focus on this atomic force microscope image here, what you see here are 3 square-shaped DNA structure, single layer DNA helix, so 2 nanometer in thickness. The size is about 60 to 70 nanometer. And what you will see is that when we add a trigger into the system, the structure starts to transform in a cascading fashion. So the structure transforms through multiple reactions. So have a multiple dynamic unit in the structure. So it works like a molecular domino. So this is one really relatively complex example of a molecular nanomachine, but how can you use such a dynamic machine? So here I choose one relatively simpler, much simpler molecular machine to give you a taste how this type of molecular device could be useful. It's really useful for making materials have tunable property, for example. It's also great for building, for example, biosensors, right? Once you have some molecule being detected, trigger response. So this is one example. We use a molecular nanomachine or dynamic device to control the interaction between 2 biomolecules. Really simple example. So the basic design of this device is just a square shape, all made of DNA, by the way. We can control the configuration of that structure to either have an open configuration or closed configuration. How do we do that? No time to discuss details here, but we can do that, trust me. So what happens is that we then can load biomolecule that we want to study on this device and control how close the distance are between these 2 molecules, because we can open or close the device as we want. So the molecule we choose as a model is just the EGFP, green fluorescence protein. So we split it into 2 halves. Many of you know that the green fluorescent protein will lose its fluorescence when you split it into 2 halves. But when you put them close to each other, they can reform into a complete structure. You will see a recovery of fluorescence signal. So that's the system we use. So we put 1 half on top, 1 half on the bottom. When it's opened, there's no fluorescence signal. But when the device is closed, you see a strong recovery of the fluorescence signal. So this is, I think, it's really simple but really great example to use the device to control biomolecule interaction. And this could be used for, you can imagine, if you want to study protein interaction and at really extremely low concentration, right? So now, because you always have high local concentration, and many of you know that for some of the protein, it's really hard to get the concentration high until they crush out from the solution. For such a system, you don't need to rely on super high concentration of protein anymore. So with that, I want to move to the second part of my talk today. This is our effort to integrate DNA nanostructure or nanomaterials with other nanomaterials, because of course the motivation I already mentioned in one of the earlier slides is you want to be able to build multi-component nanomaterials to provide more functions you want. DNA of course have its own unique property, but it's really limited, right? If you want to build a nanoelectronic device, for example, or optical device, taking advantage of plasmonic effect, DNA cannot do those. So if we can incorporate DNA with other nanomaterials, that would greatly extend its functionality. So there's different way to do that. So here are a few examples we use. So the first type of approach we use, I call it scaffolding. So we take the DNA as a template to organize nanoparticles, nanorod, protein, polymers to the configuration we want. The second type of approach I call it conversion. So we grow something on top of DNA as a template. Right? So here are an example of the 3D DNA crystal structure, which I showed you earlier. On top of this structure, we grow a thin layer, 2 nanometer layer, a thin layer of silicon dioxide to build this kind of inorganic, organic kind of hybrid material. What you see here is our couple of SEM image. So DNA basically is kind of invisible, but you can see the silicon dioxide. The third approach is a lithography method. So just like many of you know that you can use, for example, block copolymer, right, organic polymer as a lithography mask. Potentially you can use DNA as well. There's a bunch of limitations at this point. But in principle, you can do that. What if you can transfer that really high resolution, nanoscale resolution with any geometry you want as a lithography mask, use DNA, and transfer that information to the material you want? That would be great. Here is a relatively simple example of using DNA to protect it's hard to say, but it's a graphene layer so it's a graphene put on top of a substrate, and then we put a DNA structure on top of it. We have to protect DNA because DNA is too thin, so we grow some silver and gold protecting layer on top of the DNA. That allows us to use this DNA structure as a lithography mask to etch out the unprotected region of graphene. So what you end up is a graphene geometry that matches with your lithography mask. Of course, there's a lot of improvement needed to make it actually useful, but the principle works. So now if you can do all this DNA nanostructure, really cute nanostructure, I would say become a platform for you to design other type of nanomaterials which may have the function you actually want. So for today's talk, I want to kind of briefly go through a few examples, kind of provide you a little bit detail how we can transfer the structural information of DNA to different types of nanomaterials. Here, we have done a lot of work with with nanoparticles, particularly gold nanoparticle. We also work with magnetic particle, quantum dot, any nanoparticle in the right size, range. But gold particle, of course, is the easiest to work with due to the chemistry. So here is an example. So we have this kind of a hexagon-shaped DNA structure, and we want to build this, a two-dimensional array or nanotube structure with the gold particle organized in the fashion we want. So we start with building a monomer first. So we can design a DNA or a structure. This particular structure is called DNA origami. So then at the location we want to attach the gold particle, we extend a bunch of single-strand DNA or capture strands out from the surface of DNA structure. And on the surface of gold particle, in this case, a 30 nanometer gold particle, we functionalize a bunch of DNA strands that's complementary to the handle sequence or capture sequence on DNA. The reason we have multiple sequences, of course, is to be able to more precisely control location of the particle. So in this case, we can, the particle will bind to those 6 DNA strands and being precisely located at the center of the structure, as you can see in this image. Of course, you can do different configuration depending on what you want to construct. So here are some examples. We have large particle, a combination of small particle and large particle. Then to build a 2D array or this type of tube structure, what we need to do is connect the individual building block in the way we want. We can also use a similar approach to control the gold nanorod. Gold nanorod is, of course, any large aspect ratio particle is a little bit harder or trickier to work with than symmetric spherical particle, as you all know. But we can basically use the same chemistry on gold nanorod in order to, for example, put a gold nanorod on this DNA structure, which is showing this gray color underneath on this particular arm. What we need to do, again, is design complementary capture strand underneath, which is not joining here, underneath this particle. And then we functionalize the particle, nanorod with a complementary DNA and react with the DNA object. We can form different configuration, different chiral structure, and then we can, of course, connect them into higher order structure. One of the motivation while building this kind of assembly of nanoparticle and nanorod is to build chiral structure or magnetic plasmonic structure, taking advantage of the plasmonic coupling between the gold particle and gold nanorod. Here are some examples. There's a chiral structure made of gold nanorod assembly, gold nanoparticle assembly. This is a small structure with magnetic plasmonic property we want by combining this kind of 6 silver nanoparticle ring together in the configuration we want. In addition to organic particle, which we actually work a lot with, as I mentioned earlier, magnetic particle. Beyond metallic particle, we also work with magnetic particle, quantum dot particle. We're also interested in to use DNA to assemble protein, peptide, organic or biologic molecules into nanomaterials, nanostructure. Here is the example. Sorry, I keep checking time. I don't want to go over time. In the interest of saving a little bit of time, so I'm not going to discuss this original peptide here too much. So this is a collagen-like peptide that can assemble into a structure already, and we modify it. So now what happens is that we can put the DNA together with this peptide. The peptide acts as a glue to glue those DNA structure together to form this hybrid structure. So it has DNA, which is showing in this white color line here. You're looking on, from the side of this object. And in between the DNA, which you don't say anything, but there's a peptide between the DNA object. So we can directly design the DNA and peptide, in this case, to assemble into the structure, nanomaterials we want. We can also use DNA. This is another approach we use. We use DNA to guide the protein assembly. In this case, in this example we demonstrate here is using DNA to guide TMV, virus protein assembly. On a RNA strand, we put on the DNA structure, so you can assemble them into relatively intricate and complex structures. So this is the example I already talked to you about, is grow silica dioxide on top of a DNA structure. Here's just some more TEM image. One thing I do want to mention here, I'm going to skip the chemistry here. It's relatively simple two-step reaction. The one thing I do want to mention is because we can engineer the template, right? So here an example, right? We can change the, by changing our design, the DNA object, we can change the crystal structure, right? So in this case, we have a more like a cube structure. It's a kind of elongated cube. We changed how what the building block looks like and how they interact with each other. We can design, we can tune the geometry, the size of this crystal. So change the bio template here. Then we can then grow silica dioxide to convert it into a inorganic material crystal. So that's really the the beautiful part of this DNA template is highly designable. The third challenge, as I mentioned earlier, is integration of DNA nanomaterials or hybrid multi-component nanomaterial into devices. So this is a really important challenge, as you all know, is how we can bridge the gap between top-down and bottom-up, right? I'm not going to talk about our effort in this direction today. Instead, I put two reference papers here. The two back-to-back publication in Science published in 2020. Here is the author used the DNA brick structure I showed you earlier with parallel channels to anchor carbon nanotube to form nano arrays. They can control the pitch size down to about 10 nanometer. So this is really hard to achieve for carbon nanotube array with other method. This is the first step. Of course, in order to make a useful device or device with some function you want, they actually put down this device on a surface and fabricate a field effect transistor device in this case. So this is actually really challenge and direction, but really, I think it's a really important step for us moving forward to make these DNA-based nanomaterials or nanostructures useful for this type of applications. So again, I'm going to skip this our effort in this direction. So now we reached to the last part, the third part of my presentation today. So I talked to you a lot about how we can make nanostructures, how complex those structures can be, and how we can combine DNA structure with other nanomaterials, nanoparticles, proteins, to make hybridized structure with unique properties, and discussed some examples of incorporating those nanomaterials into top-down fabricated devices. But in real-world application, I think a lot of application I see actually is in the biomedical or biological research direction. It's not too surprising because DNA at the end of the day is a biomolecule. We know it has a lot of biofunctions, and it's also biodegradable. So, so this is also a really important direction in my lab, which is slightly different than the nanomaterial development. So here, what we're doing is taking the DNA structure we already have and use it for a different application. Today, I'm going to quickly show you 3 examples. So the first example is using DNA as a nano delivery system. Right? So this is really common for people work with nanoparticles or nanostructures. They often try to use it as a nano delivery system, see what effect you get in cell or in animal model. So here are a few examples. In order to use DNA as a delivery vehicle, the concept is really simple, as you can imagine. Just load the drug on your nanoparticle, then start delivery. The most easy way to, easiest drug to load, of course, is DNA or RNA-based drug. So this started first, one example here, we studied the internalization of DNA nanoparticle, how the different design affect the internalization of our structure. So we kind of get some information of certain design, for example, nanorod design internalize better than other geometry, and certain size works better for the internalization into cell. The second example, we started to do some actual drug delivery. So here we load the siRNA. This particular one is called siRNA BCL2. This is targeted BCL2, which is a protein related to tumor development. So if we can suppress the BCL2 protein expression, we can suppress the tumor growth. So this is exactly what we see in the animal study. You can see the tumor growth is suppressed when we have the delivery vehicle carrying the siRNA. This one is more recent, but 3 years ago. So we load a small molecule drug onto our DNA object, and again we can deliver into animal model. So we see a decrease in terms of the tumor development. So, so first important direction for biomedical research is using nanostructure, in our case DNA nanostructure as delivery vehicle, or for as a diagnosis imaging probe as well. The second important research going on in our lab is to use DNA to develop molecular probe. In this case, we are using DNA to develop molecular probe to detect molecular force generated by a cell receptor. I'm going to go through this part really quickly because I want to spend a couple more minutes on the last project, which probably is more clear example for you to get the general idea. So long story short, we can design DNA to have ligand and what we call the DNA force probe that can report, it will generate fluorescent signal when certain force is applied to it. So then we can put this on surface when the cell receptor bind to it, it generates a force. And looking at the fluorescent signal, we can determine how much force is generated by cell. Why this is important, partially is because the cell behavior is closely related to how much force is being generated at the interface. This is known as mechanobiology. So to be able to have a probe to understand this kind of behavior at molecular level is really useful. So we also have, in this direction, this is a really important direction, we are actually in a process of applying a center grant from NIH to further develop this type of technology. So this is another example, not going through details, but we are able to develop a probe that can give us much better resolution when we look at the force distribution on the cell surface. So now we're approaching the end of today's talk. So this is the last project I want to talk to you about. I'm going to spend hopefully 2 or 3 minutes to discuss this project. So the idea is we want to use a DNA-based system to investigate the cooperative binding between a T cell receptor and one of the co-receptors, CD4. For those of you who are not very familiar with T cell, of course, the T cell signaling and the function is closely related to the TCR receptor binding to its ligand, MHC. But this type of behavior is closely regulated by a bunch of other co-receptors, CD4 and probably more importantly, CD8. We know this through a lot of study already, but what's not known is at molecular level what's going to happen. What's really important for this, for example, one of the simple questions is, what's the distance required for this type of trivalent or trimolecular binding to happen? So we can intuitively imagine if this on cell surface, if the distance between TCR and CD4 are too far, they will not bind to MHC together. But the co-receptor, in this case CD4, only when it's close to TCR, it can have its function, can have this kind of trimolecular binding happens. So to do this, because of this, we propose a research. We can take a DNA structure and kind of generate this kind of artificial kind of configuration between a TCR and CD4. If we can anchor a TCR on our structure at one location and anchor a CD4 at another location, we can study this type of binding. The beautiful part of this is we know exactly the distance, right? So which is in no way you can control in an actual cell surface. So that's exactly what we did. I'm going to finish my talk in 1 minute. Or hopefully, or 2 minutes. So what happens is we design a one-dimensional DNA structure, and we anchor TCR, T-cell receptor on one side, and CD4 on the other location, and we tune the distance because we can do that. We can precisely design our system. This 100 nanometer separation, of course, is basically our negative control. We don't expect any trimolecular binding to happen at such large distance. Then we'll gradually titrate down the distance from 100 to 20 to 13 to 6 nanometer. And we expect at a certain point we will see a much stronger molecular binding happen. So we made this device here, a couple examples of TEM image. You can see the DNA structure down here, much larger, but you can see the tiny protein TCR and CD4 on top of the structure. This, of course, is really hard to do, but we made it eventually. And we use a method developed by my colleague, Dr. Chen Zhu, right here, using this micropipette adhesion method. So essentially, we put our DNA structure, those things, on a microbeads surface. we control the density to make sure that we have low enough density, so each time, or majority of time, we only see one DNA structure interact with the antigen presenting cell, red blood cell. So the antigen or MHC is displaced on this red blood cell. This cell is grabbed by a micropipette, as shown in this movie here. The beads is also grabbed by another micropipette. In the actual experiment, we touch these two objects close to each other. If there's binding event happen, you will see this kind of sticking event. And when you pull them apart, you will see the geometry of the red blood cell change. And depending on the how long this process is going to take and how much change of geometry we see on the red blood cell, we can calculate the stiffness of the binding or how strong the bond is. So the expectation is if there are only one TCR binding to the MHC ligand, the binding will be much weaker than the trimolecular binding if you have TCR, CD4, and the ligand. And that's exactly what we see, really great. So at 100 nanometer, our negative control, there are only one peak, relatively weak. Oh, by the way, stiffness basically is how strong the bond is. The single peak at 20, same thing. But when we move down to 13 and 6 nanometer, something really interesting happened here. We start to see a second peak, the green peak here, which corresponds to a stronger bond. We believe that's the trimolecular binding. And even more assuring is that at a smaller distance, we see the peak and it's bigger, higher than the larger distance. So essentially, we would say the threshold is below 20 nanometer, and at a smaller distance, there's a higher chance for this trimolecular binding to happen. So this is part of the important data for that publication. So this also is a good example to show you how you can use a relatively simple design to design a highly controlled system to understand a biological question. Really great example, I would argue. So with that, I want to conclude my talk today. So I want to thank my current lab member, previous members, and our founding agency. Yeah, thank you.
So we have some time for some questions. So if you have a question, please raise your hand and I can bring you the microphone so you can ask.
Great talk. So many exciting capabilities. I want to maybe hear a little bit more your perspective on integration, some of this DNA-based complex structure into functional devices and making them actually kind of truly functional and integrated. One thing I could see as challenges from the perspective, you need to align them between the electrodes, then making connections, and maybe there is all standard lithography, maybe challenging. Maybe on one device you could do it, but if you need to do 100 of them at the same time, it's now kind of going outside of normal processing. Am I reading it correctly, or this is not a big deal? And
it is. It is, of course, a big deal, right? So that's actually a bunch of questions. I think I'm going to try to kind of offer some of my opinions. So first of all, it is a big deal, right? If you have a really good way to put this type of structure down at a location on a surface, for example, at a location you want with perfect control of orientation and low defect rate of the initial structure, which can be achieved in solution, then you have a really great way to make a really good device. But that's precisely a really challenging part. In fact, Katie is here and is here. We're working with some folks here at Tech to try to address this issue. There are some already really interesting publications out there. People have used different way to control the let's just focus on the, of course, there's different type of device we can talk about. Let's just focus on, for example, putting down the structure on a solid support surface and control the location orientation. There are a bunch of research done that out there already. One method really rely on the geometry matching. So you design a object of DNA from DNA, and then you generate a surface patch or surface pattern that matches with the geometry of the structure, and use chemical modification to modify that location to have a attracting force to your DNA, and you carefully tune the condition, allow the structure to have some freedom, so the structure can adjust to the lowest energy state. It's, again, of course, it's not 99% accuracy, as you can imagine, but we are improving. Yeah. There are other methods as well. So one thing, depending on your application, for example, if we want to build the biosensor array I showed you earlier, so what you really want is some structure trapped in between two electrodes, for example. So there are different ways to do it. You can use electronic force to drive it down. You could use magnetic particle, for example, on a structure, and use magnetic field to control the orientation. So it's gigantic deal. I think if you many people are interested in this direction. There are many challenges, and there are good progress as well. I would say, yeah.
Yeah, we should probably talk about this. We are developing a tool using focused electron beam in combination with a variety of ways to deliver precursor in gas and liquid phase for essentially direct write three-dimensional nanostructures from different materials that essentially is trying to accomplish what you want to do through both topological confirmation, let's have in a pocket to capture, but also be able to make, for example, ohmic contacts. And we've done this for graphene, carbon nanotubes, and that's a big problem always, you know, and especially the standard lithography really fails in this regards. You know, all of this, spin photoresist pattern remove, it's really it doesn't work for except only one device.
Yeah, love to continue this conversation. Yeah, this is something we really want to do. Even for carbon nanotube, we're trying to make a carbon nanotube nano array. I showed you. After we made that, the biggest thing is how we control orientation, incorporate into a device. Exactly as you mentioned. Yeah, that's really really important and really challenging. Yeah.
That's great. Well, you should connect, I guess.
Sure.
Thank you.
No problem. Any other questions?
All right, thank you. Very nice talk. I want to go back to the last slides. I think that's very interesting to demonstrate the engage. But I was wondering how come you can measure the toothpick? What does toothpick mean?
Oh, sorry. Maybe like many experiments, right? This is not so uncommon, right? So what actually you say, of course, is this curve we show here is it's combination of many measurements. So for every bending event, you generate a bar here, right? And you can see a histogram here, right? So you have this distribution of stiffness or how long this bond will last. And this curve, this peak or this curve, basically is a simulation, matching, based on that data, right? This histogram down here, a bunch of single events, right? So each binding and opening generate this histogram, this light orange color down there. This curve here, matches with what we see there. In this case, it matches with a single peak model. In this, the two cases match with two peak model. Yeah. So again, this is really common, of course. So that's how we don't directly observe this peak. Is we fit our data, right, based on this histogram. Yeah.
So we have time for one more.
Thank you for sharing your beautiful work. So I'm interested in your talk, in the third part of your talk, where you describe how you use nanomaterials for drug delivery or biosensor. So I'm curious, would you comment on the stability of DNA based materials in the physiological conditions, since there are abundant of DNAs in the blood and body fluids? How do you design the DNA in a way they are stable in these conditions?
Yeah, so that's a great question, right? So for an ideal delivery system, you want to be able to control all important property, right? Stability, of course, is one of the really important things. It's not necessarily true you always want it to be super stable, right? So you want it to be stable at the right time and location, but kind of easy to degrade once it performs its task, right? So if you have a gold nanoparticle, it's super stable, but it's staying in your body forever. You don't necessarily want that. So in terms of the DNA nanostructure, when you just assemble DNA strands into a structure, it already naturally more stable or more resistant to enzymatic digestion, exonuclease, for example, than single-strand DNA or double-strand DNA. As you can imagine, just simply due to the fact you have a kind of artificial structure, a compact object, it's harder for enzyme to attack it. So those DNA structure, of course, depending on your design, could be stable in physiological relevant condition for half day, 1 day. Not super stable, but much more stable than a naked DNA strand. So that's already useful. What if you want even more stable object? I guess that's partially the question. So the of course you can chemically modify your DNA. Commercially, there are modified DNA that's much more resistant to enzymatic digestion. So you know that. You could also, for example, cross-link your DNA. So you kind of connect the ends. You don't have a 5' end or 3' end for the enzyme to attack. That's another way to stabilize. You can chemically cross-link your DNA strand to make it even more stable. You can protect it with some protein, peptide, or polymer. There are different ways to tune the stability. So all those methods are available, but of course, in reality, when you work on a particular system, you always want to start. I think the best strategy is start with a unmodified DNA and see what you see. And then if it's unstable, not stable enough, then you can start modify it, improve the stability. Yeah. So there's a lot of tools out there available. Yeah.
Okay, that's all the time we have for questions. Thank you for your excellent talk today, and let's thank our speaker one last time.
Thanks. Thanks for hosting.